Great. So just as Mike finished up yesterday with a research talk, I'm going to do the same. Deepa invited us to not only talk about broad concepts in population genetics, but also about topics that we're actively working on and some fresh results. And so I wanted to share with you some of the work that we've been doing addressing problems that arise in doing population genetic inference with next generation sequencing. And so this is going to be very nitty-gritty practical issues that arise in the work that's being done today. Before I go deeper, though, I thought I'd just give a bit of broad background about some of the problems that my group works on. As you might guess from me being here, we're interested in problems at the intersection of evolutionary biology and genetics and genomics. And we're a computational group that uses tools from theoretical population genetics and computational statistics to address a variety of different questions in this intersecting area. A lot of work that we've been doing has been around topics of genetics and geography and population stratification. So those are issues that came up a lot in the lectures that I gave in the first lecture, really. We've also worked on recombination rates, particularly estimating recombination maps in admixed individuals, inference of demographic history, both in recent human demographic history, looking at things like rates of growth and recent population sizes. And this figure here is meant to represent a demographic history of dogs and wolves, and as we've been interested in learning about the details of the domestication of dogs. And then finally, we've been interested in rare variants in humans, and so this is showing the abundance of common variants versus rare variants above the x-axis. So the common variants, and there you see there's essentially only a handful per gene when there's many more rare variants per gene as a function of the frequency spectrum that we have in humans. A lot of the work that we're doing is getting massively changed by this massively parallel sequencing. So we need to move beyond calling it next generation sequencing because soon there's going to be another generation of sequencing, and we're going to need to have a name for what we're using right now. And that is really what makes this technology what it is that it's massively parallel sequencing. You're sequencing in parallel thousands or millions of short reads at a time. And as many of you probably already know, that this is providing tremendous opportunities for biology and medicine. But of course, with all of this great promise, there are great perils. So this technology still doesn't produce perfect data. So there are a number of different problems. So one is the uncertainty in the read mapping and assembly. We get these short reads, but we don't necessarily know how they join together to represent the underlying DNA molecules that we're sequencing. There's a high per base error rate on any given sequencing read, and we get variable depth of coverage. So let me say a little more about this. I'm going to use these figures from one of the readings that I assigned, where this is from a recent review in Nature Reviews Genetics, where they show step-by-step what's happening in the sequencing. And so we have some isolate of a tissue from whatever organism we're studying, and there's some chance that there's somatic mosaicism, that there's mutations within the individual somatic tissues that might cause us to be confused. I'm going to ignore those for the rest of the talk, though. But then moving on, when we take the DNA, the genomic DNA, and we prepare libraries for the sequencing, it typically involves having the DNA be sheared into short segments, short reads, that then are amplified to make many copies of them with adapters that are ligated on. And so during these adapter ligation steps, you can have odd joinings of the short reads into what are called chimeric reads. So we might have pieces of DNA that are not actually contiguous with each other becoming joined together. We might have, during the amplification, if it's a PCR amplification, a misincorporation of a base during that step. And then we move into the actual sequencing. And in the sequencing, the way that the sequencing works is usually by a single base pair extension of millions of these reads at a time that are arrayed across a two-dimensional surface with a camera pointing down on that two-dimensional surface, taking a picture of the fluorescent light that's given off as each base pair is incorporated. And by reading off the pattern of lights in successive camera pictures of this array, you begin to see the sequence of bases that's carried in each read. So here, if we have 3 reads, and we're looking across different cycles, these 3 reads should be identical. They're actually on each of these dots on this two-dimensional surface is actually a colony of reads. They're all identical reads, and it's actually that single base pair extension should be identical across all the reads and give a single signal. But what might happen is that you get a single misincorporation of a base on one of the reads, and then that would appear as an error relative to the other ones, and we go to the next one. And so then that can cause confusion in how the signal is read. It should be reading a blue, but it might actually accidentally read a yellow, or it might read this as green, and that would introduce an error. We also might have that these different colonies of reads that should be well spaced in the array are actually overlapping each other, so that we can't identify a single fluorescent signal from those, and we end up, the algorithm then might produce garbage calls for what bases are in that location. Okay. So there's a lot that's done at this image analysis step where things could be going wrong. There's a lot that could be happening in the chemistry of the base incorporations during the sequencing, in the chemistry of the library preparation. The image analysis software that analyzes these fluorescent signals passes on to the next stage of the analysis a base quality score, a score that represents how certain at that stage that the algorithm is seeing a particular base for that particular read. So for instance, when it does have ways of picking up that there isn't a clear signal, and it can pass that information on to the next stage of the analysis. So typically where most of us begin our analyses is with this kind of data, where we have a FASTQ file, which is a version of the FASTA files that have a DNA sequence, but the FASTQ has DNA sequence for a given read paired with, underneath it, a set of base quality scores. These quality scores, they look like gibberish, but they're ASCII codings of numeric values that can be converted into an estimated probability of error. So, so specifically, in a programming language, we can convert the C into its numeric value, which might be, I can't remember the exact values, but let's say it's 3. Or I'll give a more typical score. Let's say it's 30, okay? And then that 30 corresponds to a base quality score of 10 to the negative 3. So the quality scores are, there's a multiplication by 10 and then a log base 10. So that's how you can kind of convert between what these Fred quality scores and actual probabilities of error. So for each base, so to make that more clear, if I have a quality score of 40, that would be a probability of error of 10 to the negative fourth, 50, 10 to the negative fifth, and so on. And okay, so we have the reads, we have associated quality scores, and we might see that that G that we saw in the previous example has a low quality score associated with it. Because the algorithm might have had a sense that it was a bad read. And so a theme that I'm going to get into is that we should actually be paying attention to these quality scores and not just analyzing all the sequences as if they had the same amount of information, because we actually have a hint that some of them have less information than others. Yeah. Yeah, I actually don't know the details, but it's related to things about the variance in the intensity scores, the quality of the particular signal. So it's usually a small circle. Is it perfectly circular, or is it somehow oblong that might represent it's a join of the two? So there's a bit of engineering, voodoo and trial and error and complex functions that go on there. And I actually bring up that these are guesses as to the probability of error. They're not exact probabilities of error. Yeah, the way that we recalibrate these typically is to, it depends on what system you're working on. But for instance, if you're working in humans, you have a number of sites that you're fairly certain are monomorphic, and so people will use the monomorphic sites to then, or sites that they know from genotyping arrays are homozygous, and then look at reads that have that are clearly in error because they have a difference, and then ask what's the quality score associated with that base pair, and do you see errors at those types of reads at the rate that you would expect from the given quality scores. And so you can end up recalibrating the quality scores. So that is now a standard step in analysis pipelines, is to recalibrate the quality scores and then push things forward. Yeah. Yeah, of that. Yeah. So so like in a non-model context, I think you probably would make a set of naive calls with the raw quality scores, and then take sites that are monomorphic, and then recalibrate with those, and then rerun the calls with the new quality scores, and you'd probably be in better shape. Yeah. I'll talk about settings where we're analyzing pooled sequences, and where we do a unique approach for doing the quality score recalibration. So these sequences then, they have not only the sequence of the portion of DNA that comes from the sample of interest, but they also have the adapters that were ligated on at the beginning. So we want to clip those off. And then sometimes we also clip off the ends of the reads, just because the ends of the reads oftentimes have the most error. And I'll show a little bit more about that in a second. So then once you have your reads, if you have a reference genome, you align the reads to the reference genome. And that can be problematic in that reads can mismap to the wrong locations. If there are regions of the reference genome that are highly similar to one another, then a read might not be unambiguously mappable to one location or the other. Or if the reference is actually misassembled and missing, there's a duplication in the real genome that's missing, we call that a hidden paralog, then you can have reads from that should be from two different regions of the genome stacking up on one region of the genome. That produces a particular error mode where, say that there's a hidden paralog and there's been a divergence in the duplicates, then you're going to have a set of reads that always has a difference within it, and aligning on top of the to a region where, so on average, individuals, if there's no polymorphism in the system, we just have individuals that have, they're all monomorphic for these two paralogs, but the paralogs have a divergence between them, then individuals will have a 50-50 mix of reads from these two different allelic types, and they'll be called all as heterozygotes. And I bring this up because of the little exercise that we did in the computer lab yesterday evening. You make plots of what the genotype probabilities look like relative to allele frequencies, a visual plot of Hardy-Weinberg fit, right? And in those plots, if you have sites that are from hidden paralogs, you'll see genotypes from loci that have an allele frequency of 50%, but with all of the individuals being heterozygote, that's a pretty good sign that you've got a hidden paralog in the data. So that shows some of the importance of like using basic population genetic principles, Hardy-Weinberg proportions, to clean your data and spot errors that might be due to sequencing. Okay, so I mentioned that the error rates are not consistent across the read, and one of the really strong patterns is that the errors are particularly bad towards the ends of the read. So this is error probability versus position along a read, and you can see it has this really strong increase. And here's another view of this. So if we look at the first position and look at the base quality scores, we see in the first position the base quality scores tend to be quite high. So base quality scores around 35, so less than 1 in 1,000 error probability. And then as we move along, position 15, 30, 45, 60, 75, the error rate starts to shift down into the 1 in 100 range of a base quality score of 20, and even the 1 in 10 range of a base quality score of 10. These base quality scores of 2 are the scores that the Illumina sequencing algorithms give when a base is deemed unusable. So you see more and more of these unusable reads. What's one of the major processes leading to this? If you can imagine, we're doing single base pair extension sequencing of a group of copies of a DNA molecule. As we keep going cycle by cycle, the reads start to get out of phase with one another, where maybe one particular read has extended 2 bases, another one has only extended a single one, and so you start getting you're not sequencing the same base at every time step. You're sequencing ones that are slightly off register. The most advanced image analysis pieces of software actually account for this and try to register for it, but at some point it becomes even difficult for the algorithms to correct for it, and so you get these mushy signals of what the base is and higher error probabilities. Okay, so we want to be, there are these factors. Luckily, the base qualities are given to us that give us some indication, and we can use them as a compact way to represent our uncertainty in any base call. Okay. So that's one of the main limitations. There's the idea that if we get enough coverage, that if we can get many, many reads for a single location, we can overcome some of these problems. But there are cost constraints involved here, where to get our sample sizes up to be large, for the same amount of money, we're going to have to sacrifice coverage. So if we're interested in rare variants that might tell us about recent population growth, those kinds of patterns, then we're oftentimes interested in increasing our sample size, but as we do so, we drop our coverage, and that's going to decrease the quality of our genotype calls. We also have choices that Michael mentioned coming up in his own lab about whether we pool the sequencing that we do, or whether we do special library preparations that somehow mark each individual's DNA as unique before we put it into a single sequencing lane. So we index the reads or barcode the reads with individual specific adapters that we can then later use to pull apart which reads came from which individuals. This is more costly, and but gives more accuracy. So we have these difficult choices to make, and oftentimes working with limited budgets, we're going to make choices that sacrifice coverage, that lead us to do pooling. And so what we want to talk about today is some of the challenges that arise when we're dealing with low coverage data or pooled data. Okay. You might say, well, these problems will go away. They're not inherently interesting. Why don't you, work on a problem that's going to last much longer? And I think it's true that in a technical generation or two, the sequencing is going to be much higher quality, and these problems will be diminished. But I think there's still going to be extreme users. So people with groups that are working with particularly constrained budgets, or who are particularly ambitious in terms of, even when the sequencing gets better per read level, where the accuracy gets better per read, you're still going to have groups that want to get the sample size up and do that at the cost of lower coverage. You can always push the limits, and so I think there'll still be these problems are worth solving because they're going to stick around for at least a set of extreme users. And then, quite honestly, these are problems that are facing us today, and we want to answer biological questions today. We have data sets that we've gotten, and we need to understand if these sequencing errors are going to be misleading us in our analyses. And so so we've gotten our hands messy and tried to solve these problems in our own work. So the context where we've been thinking about these problems are in the domain of human population genetic studies, where we do have studies we're involved in that have large sample sizes and low coverage. So my first exposure to this was a extremely large sample size study, one of the largest sequencing studies done in any species to date, that had 14,002 individuals sequenced for 202 genes with 27x coverage. So people say the rule of thumb is the 80x coverage is where the sequencing errors are really something you can ignore. But at 27x, are we okay? We didn't actually know when we began the work. We're involved in another study now that's looking at around 2,000 individuals with 4 to 6x data. To get those numbers up, you have to make sacrifices in terms of coverage. We do this because we're interested in rare variants that might tell us about the recent past, that might be potentially deleterious and only transiently in the population at low frequency. So that's one domain. Another domain is in the wolf-dog whole genome sequencing that we've been doing, working on understanding domestication and studying sites under selection. And there, we were able to raise funds and get reagents to do 6 canid genomes, so these are mammalian-sized genomes, but to get those 6 samples that we wanted, we could only do them about 20x coverage each. And so is that enough? Is that problematic? And then finally, and this is one area I'm going to talk about quite a bit today, are these Evolve-And-Resequence studies, where I'll elaborate on them in another slide or two, but the question is about how to analyze pooled sequencing data. So so specifically I'll talk about these haplotype-based analyses of Evolve-And-Resequence experiments. This is work that my PhD student, Darren Kestner, has been doing as he's getting towards finishing his PhD, and inference of frequency spectra with low coverage data. And this is work of Yunjong Hong, a PhD student. It's also getting close to finishing a PhD. So, so let's launch into this first project, which regards these Evolve-And-Resequence studies. So the goal here is to map the genetic basis of complex traits. So as opposed to the experimental evolution studies that Michael was telling us about, where the interest is in the process of adaptive evolution, the community that's using Evolve-And-Resequence studies, mainly in Drosophila, are oftentimes interested in just the genetic basis of the complex trait, a complex trait that has variation in the starting lines. And so where if we just tried to do a a genome-wide association looking at this particular phenotype against looking for associations with given genotypes, we might not have enough power. And so the idea is to take a starting population, and in our work with our collaborator Tom Turner on this, he starts with 162 founder lines that are inbred, and they're fully sequenced. And these are from the Drosophila genetic reference panel. And then he, in the lab, allows them to outcross and mix freely. And then he selects for different lines in different directions for extreme values of whatever trait is of interest, and does this for several generations on the scale of 15, 30 generations, and then does pooled sequencing, where he takes representatives of the high line, representatives of the low line, and grinds them up and puts them on a single lane of a high-seq machine, and then makes some estimate of allele frequencies, or we could use haplotype frequencies, and compares them across these different pools, and tries to find regions of the genome that have changed in frequency, because those should be the QTL that underlie the trait in the founder line. Okay? The time scale is not enough here that one is expecting new mutations to arise. They may, but what should be shifting in these first generations is the standing variation that exists amongst the founder lines. There's no reason one couldn't, think about studying it that with looking at novel mutations. But I'd say for the analysis methods that we're about to show, they're particularly suited for looking at variants that exist in the founder lines. Okay, so these approaches, they're suited well for lab-reared organisms with short generation times, and they're becoming increasingly popular. Tom's work in this area has focused on, so far, on the complex trait of courtship song. In particular, he's looking at the interpulse interval between these pulses in the songs of Drosophila, and then trying to map the genes that underlie this, because if you could understand this, you could understand the genetic basis of some of the characters that are sexually selected and used in mate recognition and so forth. So this IPI in milliseconds in the founder line had an average of 35, and these are in 2 replicate high lines, 2 replicate low lines. You see a nice response to selection, which suggests there's standing variation for the trait, and that it's a heritable trait, and presumably that the allele frequencies underlying it are shifting, and that then if we sequence these 2 lines and compare allele frequencies, we might learn about what loci are involved. So this is a plot from his original analyses. It's a little tricky to get, but what you see in blue this blue cloud are comparisons of the short versus the short line and the long versus the long line on the Y, short versus short on the X. And then in the red cloud, we have short versus long for one set of replicates, and on the other axis, short versus long for the other set of replicates. So locations here where the same allele increased in both replicate short versus long lines are good candidates, or ones over at this corner are good candidates for being QTL for this trait. Combining these results with the genome-wide association results from looking just in the founder lines, Tom's been able to find that one of these loci is the syntrophin-like 1 gene. So he's made some progress in learning a locus affecting this trait. And so this work and work by other groups has started to create a lot of interest in this area, and maybe use it as a inexpensive tool for mapping traits efficiently. So what's involved here is keeping the fly lines, doing the selection experiments, which is he's been able to do with undergraduate students interested in getting some exposure to working in fly labs, And then, per trait, he's just done 4 lanes of Illumina sequencing there for each of those replicate lines, which is approachable, much more approachable than trying to do a large-scale genome-wide association study with many more lines. Okay, so how do we best analyze this kind of data? So you can do a vanilla approach of just taking the stack of reads that you have and counting the bases in the sequences to get an allele frequency for each base at each site. Okay. And that's what was used for the plots that I just showed you and in Tom's initial work in this area. But as I was discussing this with him, to recognize that there's a lot more information in here. So you have, first off, you have the known founder haplotype sequences, because the study began with 162 lines that were fully sequenced. So you could use that to help guide the interpretation of the data. And then the reads themselves are long enough that sometimes they actually have multiple mutations within them. This wouldn't be the case in a species like humans, where the effective population size is too small to have polymorphisms at such density. But in Drosophila, you actually will get multiple mutations within multiple variant sites within a single read. And so you actually have quite a bit of haplotype information in this experiment. And then, so that's the point. Reads cover multiple variant sites. And then you also have this useful information in the base quality scores that if you just count bases, you're not taking advantage of that, and you're biasing your frequency estimates because you're counting errors as real bases. So we wanted to then work on a method that would be a base quality aware approach that could infer, instead of allele frequencies at each location, haplotype frequencies, and in that way gain more information about the local genetic variation in each region of the genome. So this is really a statistical genetics problem, and it's a fun one. So this is a schematic of the problem. So if we have these known haplotypes at the top from the 162 sequenced lines, we then have to think about how we have some underlying true haplotype frequencies. So I have the stacked bar chart to represent that. Let's suppose in the pool of sequences that there's about 50% of this pink line, about 25% of this blue, and 25% of this green. Then we get reads that are generated on the basis of these underlying haplotype frequencies. And the reads, in some cases, may be perfectly identifiable as being from one haplotype. Ideally, it'd be perfectly identifiable as coming from say, the green haplotype. And then maybe here, these carry two alleles that are only found on the blue haplotype, so we can get those definitively to there. But there may be some ambiguity where some reads we can't be sure which haplotypes they come from, right? And so we somehow want to work with this, this data to get to estimated haplotype frequencies. Right? So we could think about naive approaches of doing this, where we say, oh, if this matches these two, well, let's give it a weight of a half to this haplotype, a weight of a half to this haplotype, or let's just ignore it and only use the reads that are ambiguously matching to haplotypes. But we can do better, and we can do better if we know one thing, which is if we could know what the haplotype identity of each read is, then it would be a simple counting exercise, right? So if you told me that this is definitively from green, then I could count them up and move forward. So this is it's another case like in the structure model that I showed earlier, where we didn't know the ancestry of each gene copy, but if you add that variable into the model, then a lot of the probabilistic calculations become straightforward. We have a similar latent variable here that if we know the underlying haplotype identity, it's quite simple to estimate haplotype frequencies, right? The problem comes up in the ambiguities. So we're going to introduce that variable into the model. Okay? And I'm going to show some equations in the next few slides, and let's just try to think about this. This is a probabilistic model for this data that is going to help us see how we can get to doing some good inference in this problem. So we have 5 major variables here, and I'll walk us through them. They're all represented in this graph. So at the top here we have the frequencies of each of the founder haplotypes. That's the thing we're really interested in. And the frequencies are going to determine what the reads have. So this box here is representing a set of n reads that we have in our sequence experiment. And each read has associated with it this z variable, which is an identifier of which haplotype the read comes from. And so if I have, say, a 50-50 mix of 2 haplotypes, then that tells me that my z variable for a given read has a 50% probability of being the first haplotype, 50% being the second. Right? And then if you tell me what haplotype a read comes from, and some other information, I can tell you what you would see for the data. So what's that other information? The other information is I need to have a database of the original sequences for the 162 founder lines. That's here in X. Okay? And then I have the quality scores for each of the reads and each base in each read. So now this inner box is for each read, we have another set, the set of bases for that read. And so now I can tell you what I will see as the exact base at each position in each read in a probabilistic way. If you give me the quality score, you give me what the true base should be, and you give me which haplotype I should be reading off of, then I can say exactly what I'll see here. Okay? So the components of this model are the haplotype identities for each read, which is just a multinomial draw from the frequencies. And then for each base, it's going to be this probability distribution where it's going to, the base is going to exactly equal what the haplotype, the sequenced founder line has if we have 1 minus our error probability. And if with an error probability divided by 1/3, we're going to make a mistake to one of the other bases that is not the base that is in the haplotype, the founding haplotype. Okay. So it's right now, yeah, it's a model where errors go equally in all 3 directions. Now we could make this more advanced by taking into account that the errors actually are known to be. There are certain error modes where some nucleotides are more often mistaken as others, but as a start, we just use a simple error model in this way. Yeah, so that's right. We're not. You'll see this model doesn't have recombination in here because we're going to be looking at haplotypes computed in windows that we think are short, like 100 kb windows, and where there's a small chance of there being a recombination. If there is recombination, then what will happen is you end up putting weight on both of the founder haplotypes. So that's an extension that we want to explore, though. But yeah, this version just has no recombination. So okay, so this is a classic type of inference problem where we have a single latent variable that we need to integrate out to connect our data, what's observed, with these gray shaded out areas, and the thing that we want to infer, which is up here, the frequencies. And so it's very natural to use an EM algorithm for this type of problem. And I know these equations look a little scary, but I want us to walk through them and try to overcome these fears. So the EM algorithm ends up being it's an iterative algorithm where we take our estimates of the we start with some initial guesses of the frequencies. So we can start out with a guess that's from the uniform probability over all sets of vectors that sum to 1, and then update that estimate through time, and eventually the estimate should converge. It should stop changing, and when it stops changing, it should be at least a local optimum of the likelihood surface, and it should be we can use it as our maximum likelihood estimate. If it is a local, we can actually start if we're worried about that local optimum problem, we can start from multiple locations, and hopefully all of them will converge on the same location. Okay, but that's a side note. Okay, so we have the frequency for the kth haplotype at the t+1th iteration of our algorithm is going to look like the following. It's going to be an average of these expectations of the indicator variable that a read comes from the kth haplotype. So we're essentially saying, how often do reads come from haplotype K? That's what our guess is going to be of the frequency of K. Okay. So what is this expectation right here, though? Let's look at it. An expectation of an indicator variable is just the probability of that variable taking on a set of values. So this is the probability that the rth read is from the kth haplotype, given all the other information in the model. And that breaks up in this way, and this is really where we want to focus on. There's a denominator here that's not very important. It's just a normalizing constant. And then these two terms we want to look at. So let's focus on this part right here. This is a product over the bases where we compute the probability of the base given which haplotype it comes from, which quality score it has, and what the founder sequences are. So this is really just the error model. And then here we have in front the probability that the rth read comes from the kth haplotype given our previous estimate of the frequencies. Okay? And this is a really important step because it actually helps us resolve when a read doesn't have very clear information. So say that a particular read has only some 50-50 probability of coming from haplotype K and some other haplotype. If we already know from our previous iterations and looking at and essentially looking at other reads that haplotype K is very abundant in the sample, then this term is going to push that read towards being haplotype K, instead of giving it equal weight of being from those two haplotypes. And that ends up, in a very principled way, pooling information across the whole sample to carry out the inference and refining the estimates substantially. Okay. So we have a base quality aware approach here. It's taking a product across the whole read, so it's using read level information. It's using the founder haplotypes and the X's there, and then it's pooling information across reads, which gives us another advantage. So how does this method work in practice then, and in comparison to other methods? So we've done some simulations using 100 base pair paired end reads, 200x coverage of 20 haplotypes, founder haplotypes, and then looking at some competing methods. So there are methods that work with where you first estimate allele frequencies and then use the allele frequencies to estimate the haplotype frequencies. One of the best of these is a method called HIPPO from Purinan et al. In 2009. And then we also try two non-allele frequency-based methods, one that we call string match and the other one which we call soft string match. And the string match method, what we're going to do is assign a read to a founder haplotype if it's a perfect match, and then just sum over those assignments to get frequency estimates. It seems like a pretty straightforward thing to do. This is kind of analogous to doing if we were to, it will come up later, like do, blast alignment and then, take all the hits that have a perfect match and then, count our abundance of a species based on the number of perfect matches. But we also might want to allow some imperfect matching. So that's the soft string match, where we allow some error in the matching the haplotypes. And so this is the sum of squared errors in a measure of how distant our haplotype frequency estimates are from the underlying haplotype estimates. We have true values. So we've simulated data where we know the underlying haplotype frequencies, and then generate read sequencing reads with error, and then apply these different algorithms, compare our estimates to the truth, and see what the error rate is. And so you can see here for these are 500 base pair windows, 1,000 base pair windows, going up to 5 KB windows, that our errors rate is much lower with the EM approach than with the soft string, the string match, or the allele frequency-based methods. As you get to larger and larger windows, the allele frequency-based method becomes computationally infeasible to run, so it drops off. And you can see as you get to bigger and bigger windows, these haplotype frequency estimates get extremely accurate relative to the alternative approaches. So that's great news. How do other factors affect it? Affect it in pretty straightforward ways. So if you increase the coverage, the behavior increases. You already saw if you increase the window size, going from 25 KB to 400 KB, the performance improves. We get lower error rates. If the read length is longer, we get lower error rates. And if the read error rate increases, we get higher error rates. So those all make sense. But we have a sense now of how we can do under different conditions for the experiment and what kind of cost you get if you lower your coverage from 200x to, say, 100x. Okay. There was the issue of should we actually trust the base quality scores? So what's shown here is if we realize that what we could do is use sites in the founder lines that are monomorphic. And if we see any sites in the reads that disagree with those are most likely errors in the sequencing. And so we can use those to get a empirical measure of the quality of bases. And we can get that error rate as a function of the reported base quality score. And so what do we see? We see if the reported base quality score is, say, 40, so an error rate of 10 to the negative fourth. In practice, the error rate was actually a little bit greater than 10 to the negative third. So its empirical quality score was something more like 25. So reported high-quality bases were actually oftentimes lower quality than reported. And bases that had very low quality scores, oftentimes in practice, actually had higher quality. And so, so we can actually then use this relationship to recalibrate our quality scores. And when we do that, we see about a twofold improvement in the performance of the algorithm. Okay, so those quality score recalibration steps really do help a lot, and are important to take into account. Okay, so how does this actually help when it comes to these evolve and resequence studies? Well, if we're taking an allele frequency-based approach to doing the identification of regions of the genome that have changed and that have somehow changed during the process of the experiment, we can take the haplotype frequencies for any window and collapse them down to get site-by-site allele frequencies, because for every set of haplotype frequencies, there's a set of allele frequencies that must hold. And so that's what we've done here. We've collapsed them down to single-site allele frequencies, and then used the same kinds of statistics that were used in Tom's original paper to try to identify the QTL. And what I'm showing here are power curves, so power's on the Y, for different effect sizes in each panel. So these are for traits with 10 QTLs, a total heritability of 0.5, a focal QTL with a frequency of 0.2% in the starting population, and the effect size given. So 0.04, 0.06, 0.08, 0.1 in terms of that effect size is in units of the standard deviation of the environmental variance. So in quantitative genetics, you have some residual environmental variance, and we can measure effect size relative to that for a single trait. And what we see is that in cases where there's like a large effect size and it's relatively easy to map the trait, you have high power over even stringent false positive rates, you get, it doesn't really matter too much which method you use. But if the effect size is smaller, where the power is more limited, so we can focus on this case, the blue line is what you get if you use the vanilla algorithm of piling up the reads and just counting the bases. The red line is what you get if you actually had access to the true haplotype frequencies. And the green line is what you get if you estimate the haplotype frequencies using our approach. So we're basically recovering, we can't improve the power better than knowing the actual haplotype frequencies, but we've at least, removed the issues that arise with having all the next-gen sequencing involved. Okay, so so there are parameter regimes where this really can help in terms of detecting sites that might have weak effect sizes. So yeah, so we're quite happy with that, but one might also ask, can you do better by looking at the haplotypic patterns? And so these are, this is a plot of the haplotype frequencies where we now have stacked up the 100 colors for the 162 founder haplotypes, and we're looking across a single chromosome arm, and we can see regions where these two founder haplotypes have increased in frequency rather dramatically over a very large window, suggesting there may be some very large effect mutation that is moving really dramatically during the selection experiment. And so we might imagine using haplotype-based statistics and these haplotype frequencies to understand these patterns of selection in more detail. Okay. This is a case where it's a possible soft sweep where two of the founder haplotypes carry the trait increasing allele and have been pushed up in frequency. Okay. So now I want us to, if there are any questions at this point, I could take them before I move on to a slightly a closely related problem. I'll also take questions at the end. Yeah. No, so it's actually completely uncoupled. The base quality score is at the level of the read, and the coverage is about how many reads you sequence. So, yeah, so they're uncoupled. If you have, because base qualities are not that high in practice, they're somewhere between 1 in 100, 1 in 1,000 in terms of the error rates, then we strive to get high coverage to make up for that, but there's no inherent coupling of the two. Yeah. Okay. So so yeah, now I want us to consider this related problem, which is the inference of microbiome community composition. So there's a lot of interest in microbiome research these days, and one of the central problems is how to identify the relative abundance of different species or taxa within your sample. Okay, and so you're given short sequence reads from a particular microbiome sample, so reads that come from, say, the gut, and you have reference reads from known taxa. So if it's a well-studied microbiome like the human gut, there's a relatively well-known set of taxa that are present in human guts and that we have some reference sequences for. And the goal is to infer the relative frequencies of those taxa. So we might be interested in knowing for different individuals the fraction of the sample that is of Firmicutes versus Bacteroides, because that has a lot of influence on the health of the gut microbiome, for instance. And once we have these frequencies of the different microbiome taxa, we can do quantitative description and comparison of microbiomes and see what kind of factors are affecting these distributions. The approach that's usually taken to do this is to do PCR amplification of the 16S ribosomal DNA and pull out a particular focal amplicon region that is then sequenced. And then what is usually done is to take the reads and I'll blast search them against the set of references. And if there are 97% sequence similarity between the reads in the microbiome sample and a particular reference sequence, then call it as being of that taxa and count to get the relative abundance of that particular taxa. Okay, so some of you might be seeing this sounds a lot like what I was talking about a few minutes ago. There are other approaches that are based on using 8 base word abundances and sequences, where those 8 base word abundances are, they also ignore the base quality scores. They just look at the raw FASTA part of the sequence, not the so you know, we have this challenge then that, you know, next-gen sequencing data have non-trivial error rates, so this approach may suffer from problems. This 97% sequence similarity, if you're treating all bases as identical, you might trip over the threshold because of some low-quality bases. So when we think about this problem, we realize it's a different problem, but it has the same underlying probabilistic structure, the same graph. So we have frequency of taxa in the microbiome sample. We have a set of n reads that each come from one of the taxa, that each have some taxa identity. And if you told me the taxa identity, the underlying reference sequence, and some quality scores, I could tell you the probability of observing particular bases at that location in a read. And so the same EM algorithm works for this problem. Okay. And this actually makes clear why sometimes it's quite useful to use these graphical models to think about probabilistic models, because you start to see themes in the structure of problems that carries out and reappear in different contexts. Okay, so we then can apply the same EM algorithm to this problem. We have slightly different types of parameter settings. We're now in a lot of these settings, people use shorter read lengths that are single end, and we're going to do some simulations to see how well we do in this problem. We're using error rates typical of the ones that are seen in the microbiome, human microbiome project mock community data, and we have we have to do read alignment to all references before we begin our computations. So we've looked at 2 scenarios. One is the Human Microbiome Project, which is a large multi-institution grant, NIH-funded project, assembled a mock community where they spiked in a bunch of taxa at different frequencies. It was 21 organisms, and sequenced them, and then it made that data available for testing of algorithms. And then we're also going to look at a typical microbiome, one that's, because this would be a low complexity microbiome in practice, because most microbiomes have more than 21 taxa involved. So a more typical one where we have 200 taxa chosen at random from the 200 taxa chosen from a larger set of 1,000 that are each from the green genes reference. So we might imagine there are 1,000 possible taxa you might see in the gut, but only 200 might be in one particular sample. And so we're going to be interested in estimating the frequency of those 1,000 and hopefully seeing, you know, only non-trivial frequencies at the 200. And we're going to compare ourselves to the QIIME pipeline, which is this very well-used pipeline in this arena of work, using the default clustering level of 97% sequence similarity, although we'll also play with some other sequence similarities to see how they do. And so how do we do in practice? Our approach, which we call HARP, for haplotype analysis of read pools, is shown here in blue, and then QIIME with the default and with the softer levels of thresholds of 0.9 and 0.8 are shown here. So we get lower error rates with our approach, and this has impacts on the Shannon diversity index. So one way in which we oftentimes are interested in describing microbiome communities is to talk about the diversity of that community. And so in gray, I show the actual diversity across the distribution across multiple samples, and then the distribution that we get from using HARP, and then the distribution from using variants of the QIIME pipeline. And you see that because the frequencies of different taxa are being estimated incorrectly, on average, it's taking reads that have errors in them, and they're matching to incorrect taxa and inflating the total number of taxa on the sample and leading to inflations in the diversity that are not, necessarily because of underlying diversity in the sample. So you can imagine this might be an issue, especially when you start comparing microbiomes from different levels of coverage, because the differing levels of error are really going to influence how much QIIME does this overinflation. You might say, okay, well, what if we zoom out and look at higher levels of taxonomic of taxonomy? And so QIIME has a way of breaking up the taxa in the reference set into genus level classifications, family level, order, class, and phylum. And you can see, so this is genus level going over to phylum level, that as you go outwards, the relative performance of using QIIME with relatively loose thresholds improves pretty dramatically. But the more principled approach that using HARP consistently is doing well. What are the... Yes. Yeah. Oh, how different are the frequencies between those? Yeah. Yes. Yeah. So I don't have a number, but we did explore how the so we're drawing frequencies in the simulations from Dirichlet distributions, but from that we can tweak how much entropy there is in that distribution, about how, whether it's relatively uniform, all of them being equally represented, or there's a few that are very abundant and then many that are rare. And you do better when it's more uniform than when it's more rare. And I can't remember the exact values that we used for these simulations, to be honest. Yeah. That for some of the simulation settings that we used, yeah. So yeah, and I can't remember what, it's some intermediate value here, but I can't remember what it is. Yeah. Yeah, that it's going to be some. Now, I mean, maybe we can get a sense of it. I mean, for the Shannon diversity of 2 relative to throwing, well, this is from the 21 taxa example, but I don't have the diversities. Oh, I do here. Yeah, diversity range of 4 to 5 for 200. for 200 taxa. Yeah, that gives you a hint, but yeah, it would be nice to have, like, let's just show what some of the frequency distributions look like, and so that you'd have a better sense of it. I think if we looked at I could find results offline later if we want to look at that in more detail. Okay, so let's move on. This thing's doing well, and it does well even on the more complicated problems. So not just on the mock community, but on when you have the more complicated scenario with 200 taxa out of 1,000. Okay, so now we've got better accuracy, but at what cost? So the cost is about 9-fold. So you can break up the steps in terms of how much time they take, and using we have to spend more time in doing the alignment of the reads, because we align all reads to all references, and then there's a part of HARP that you pre-compute all the likelihoods of reads coming from particular references, and then once you have that, you can do the EM algorithm relatively quickly. So the whole thing takes 43 minutes for 100 replicates runs, or sorry, average per run, whereas Chime would take 5 minutes. So you might say, well, look, I've spent a lot of money on this data set. I don't mind taking 9 times longer to run the algorithm. This is a bit conservative, though, I should say, because these first 2 steps easily distribute on a cluster. So if you have access to a cluster where you can do, run things on 10 nodes, you can, get that back, that tenfold, roughly tenfold cost. Okay. Yeah, so this same framework of analyzing haplotype pooled sequencing data and inferring haplotype frequencies works well for the Evolve-And-Resequence problem. It also works well for the microbiome community composition problem. And then now to finish up, I want to talk about inferring frequency spectra with low coverage data. So the frequency spectrum, as you were introduced to and came up multiple times during this workshop, is the number of sites that take on each of several different frequencies. So in a sample of say 10 chromosomes, 10 gene copies, we have possible frequencies from 1 to 9 of a derived allele. So these would be low frequency variants here, high frequency derived variants here. And the shape of this tells us a lot about population genetic processes, either demographic processes acting across the whole genome, or effects of selection. And it's also, in many ways, you can think of it as a sufficient statistic for many other kinds of computations. So from the site frequency spectrum, you can compute to GMAS D, you can compute Fay and Woos H, you can compute nucleotide diversity. So if we can get this correct, then we can get other downstream analyses correct. So how we actually go about inferring the site frequency spectrum using next-gen sequencing data? Well, we can start with the sequence reads, create genotype calls. And there are 2 major ways of getting from the reads to the genotype calls. One is to use a single sample caller, where you look at just the reads from a single individual and make the calls. The other way is to do multi-sample calling, where you might have a batch of 100 individuals or 10 individuals, and you call all of them together. And those multi-sample approaches are pooling information across the whole set, trying to use knowledge about Hardy-Weinberg equilibrium and to help refine the genotype calls. And that can help in, for instance, if there's a few reads that in one individual that might make it look like a heterozygote, but the rest of the sample is saying that it's monomorphic, then you might be more willing to believe that those few reads are actually just errors and not true evidence for heterozygote. So there's those 2 ways of doing the analysis. And from the genotype calls, then you can just do simple counting to get the frequency spectra. Or you could imagine doing a direct estimation, where you estimate the frequency spectra directly from the sequence reads, essentially averaging over all the possible genotype calls with the underlying uncertainty that you have in those genotype calls. And this direct estimation there's a nice algorithm for it developed by Rasmus Nielsen's group that's included in their software package, Angst. And you might guess that if we can think of the genotype calls as being missing data, that it's another situation where the EM algorithm becomes useful, and indeed this approach uses an EM-based algorithm. Okay. So, simple question here: how does the choice of method impact results? We're doing our analysis, and some of our work is we're actually using this pipeline of going genotype calls to frequency spectrum. We want to know, is this really problematic or not? And so what I want to show you is at different levels of coverage, 2, 5x, and 10x, what you see when you use the single sample callers, the multi-sample callers, or the direct inference approach using the Angst software. And we see is the single sample callers relative to the true frequency spectra, shown in gray here, we on average will get an excess of singletons. And I should say there are two major methods used for calling genotypes with next-gen sequencing, SAM tools and GATK. And so these single sample callers will overcall singletons. The multi-sample callers pool information and are going to be less susceptible to making those low-frequency errors, or errors of calling low-frequency variants in excess. But they actually overcorrect, and so they undercall the number of singletons. And if we use the direct inference at 2x coverage, we actually do well with getting the frequency spectra. At 5x coverage, we still have the problems of excess singletons. In the single sample callers, a deficiency in the multi-sample callers. At 10x, those problems start to go away. And so this makes us a lot happier that when with some of the projects we've been involved in that have 20x coverage, 27x coverage, and we've been taking the more vanilla approach of calling genotypes and then inferring the frequency spectra, that we're probably okay. But in some of our ongoing projects where we have 5x data, we're now saying, okay, we're going to need to scale up and use that Angst software, deploy that to get good estimates of the frequency spectra. These differences have a major impact on our inferences. So if we ask how well we infer recent population growth using the DADI method, that diffusion-based method that I mentioned last lecture, let's just focus down here on the lower right. These are the kinds of frequency spectra we get if we simulate a case with population growth. These are our estimates, but let's just focus here. These are the trajectories through time of the population history that we reconstruct using each of the different methods. So if we're at 2x coverage and we have a multi-sample calls from SAM tools, we'll actually infer that the growth was relatively weak and that the ancestral population size was quite large. If we have the single sample callers, we'll infer that the ancestral size was small and the growth was really strong. And if we have the direct sample, the truth here is in black, and the direct is sitting right on top of it, which is nice to see. And then, yeah, like I said, the single sample callers deviate in one direction, and the multi-sample callers deviate in the other. In fact, the multi-sample with GATK is actually inferring a population decline. That curve is actually saying that the population was bigger in the past than it is today, when in fact it was smaller in the past. So you get a major impact from which pipeline choice you decide to use, SAM tools or GATK, single versus multi, when you're in this low coverage regime. As you get to higher coverage, it doesn't matter very much what you do. Okay, so that's a key lesson here. When you're doing selection scans where you might slide along the window looking at relative values of Tajima's D, it actually turns out not to matter too much because you've biased yourself, like with a multi-sample caller, you bias yourself towards positive Tajima's D, but you do so relatively in equal ways across the whole genome. Likewise, with single sample callers, you bias yourself towards negative values, so you do, the same way across the whole genome. So your relative ranking of regions doesn't change very much. And so that's what's shown here. You see, here's Tajima's D across the different approaches, and you can see how the multi-sample callers in the blue shift up towards positive, the yellow and green, the single sample callers shift towards the negative. But when we look at the relative, just percentile of each window, they're basically aligning on top of each other because the ranks aren't changing very much. Okay. You get a slight boost in power if you use the direct estimation, but it's not very much. Okay, so this is a good thing to use this direct EM-based method. So then why not use it all the time? Why haven't we used it on all of our samples so far? Because it's very computationally expensive to run. Okay, and so when you get for just a single 100 KB window, as you get up into large sample sizes, it can take tens of minutes just for a 100 KB window. So if you're analyzing genome-wide data and are trying to get frequency spectra in a sliding window, that can be really troublesome. So I'm going to skip over the details of this next part, but basically the EM algorithm has this part in it where it's computing a large triangular matrix, where if you look at most of the time, the parts of the matrix that have any non-zero values, which are what's shown here in the colors, are concentrated in just a section of the matrix. And so we can take a bit of a hack and say, you know what, let's not compute this part of the matrix unless we really have to. And if we do that, so we're kind of chopping this off and just focusing on this area, we can actually take the runtime from the original and make it a lot more efficient. So now we're going linear in the sample size in terms of our computation time, and we've cut it quite dramatically. And so with these kinds of speed improvements, we can actually imagine applying this at a large scale. So this is just to say that when we take this hack, it actually doesn't affect the results very much. We get essentially the same results as if we didn't done it exactly. And so we can, then try to apply this for some of the data we're working on where we have really large sample sizes, full genome data. This is a study that we're doing with Sardinia. So these 5 Italian individuals in that map of Europe PCA, they actually, if we merge data sets, actually the human genome diversity panel data set that was in the computer exercise from yesterday, if we merge that with this data set, the HEDP Sardinians all land over here. So these 5 Italians are from Sardinia. There's a lot of interesting reasons why one might study Sardinia. And but yeah, to analyze this data, we're going to be using these algorithmic improvements. So okay, so that's what I wanted to talk about. These nitty-gritty problems, but I think they're important ones. They're the kind of problems that we face as we get into this sequencing data, and hopefully be able to learn about the kind of reasoning that's going on as we try to squeeze as much information out of this data as we can, which then allows us to get away with lower coverages with pooled sequencing data. And we're paying attention to algorithmic aspects because they really can become bottlenecks in the analysis. So there's this really interesting blog post by a computational scientist, Glenn Lockwood, looking at the improved capacity of some of the sequencing technologies, and just in terms of raw dollars, what it will cost for the storage of all the data, for the computation time, and he says in that post, I'm nervous that the new era will really be one in which the net rate of discovery is finally not limited by sequencing technology, which is what we always talk about. Oh, if I could only sequence more for cheaper, but by available computing capacity. Will it be, do I have the money to pay the server farm to handle the data? So that's why I think that it is really important to pay attention to these issues. And then to not end on a pessimistic note, this is a quote that I love from James Crow, which is, you shall see with a little ingenuity, and I think that's the key part, with just thinking a little bit about the structure of these problems and applying some ingenuity, we can do a great deal of genetic analysis in natural populations despite our inability to carry out controlled experiments, right? Despite our ability to get perfect sequencing data. We really can get away with a lot. So I want to acknowledge my lab group, the group that I've had at UCLA for last several years, and then I've recently moved to the University of Chicago, where I'm starting a small group. So I've traded the West Coast for the coast of Lake Michigan, the third coast of the United States, and especially Darren Kessner and Yinjong Han for their work, Tom Turner for the collaboration on the pooled sequencing, and Janet Sinsheimer for advising also on that site frequency spectrum project. And then I think I'm the last of the official lecturers, so I'm going to, for the sake of the whole group, thank you for your attention. This is from our rickshaw ride last night, which was quite fun. And on the way back, we had two rickshaws where the driver seemed to be in a competition to get us to the hotel first, which made it all the more exciting to ride in it. And no, but yeah, I think I'll try to speak for the other lecturers at our meals. We've been saying what an impressive group you guys have been in the level of attention and the quality of the questions that you've been giving. So it's been really a pleasure to be here and to get to know you better. And I guess that's going to be taken to the next level in our student presentations next. So great. So yeah, let's have some coffee, and I'll look forward to those presentations. Thanks, guys.